guys, for example, developed the mesoporous particles, and in 2010 it showed about 5 square meter per gram. Just comparing it to those rutile nanoparticles, it's about 170, and yeah, it's an advantage for improving the capacity by using those nanoparticles. So the aims in this study was first to initially testing the column for its ability for binding phosphopeptides. First, a simple synthetic peptide of phosphorylated peptide was used. The next step was using a more complex sample, which was standard protein, protein alpha casein, which is known to have several phosphorylated sites in the sequence. And the second part was to compare 3 columns for the binding ability of a complex sample, which again was either digest. And what was compared was, of course, a blank column, so the monolith, containing nano-titanium dioxide rutile particles, and also anatase titanium dioxide, which is also a commonly used material. So the column this is the column as it looks. It's a 100 microliter column, and I will jump to the next. Yeah. So this was simple testing for the column. The titanium dioxide column using this phosphorylated peptide, and also non-phosphorylated, and it showed in the loading phase without phosphorylation it goes through the column, it does not bind, and here it shows that the binding was okay. So the next experiment for those 2 types of samples, this phosphopeptide dimensional procedure was performed according to a paper from Manzanek from 2010. So first, after therapeutic digestion of the samples, the sample was loaded on the column with a very acidic and high salt concentrated loading solvent. The flow-through was collected. The next step was to wash with, again, the loading solvent. And after a second wash was performed with high content, so 80% acetonitrile. And after that, elution was performed with ammonium dihydrogate phosphate at pH 10.5. Those 3 fractions first were evaporated to remove acetonitrile from here and to concentrate the sample. After acidification of the elution fractions, so all samples were introduced to a CMS in the second dimension separation. And here you can see an example chromatogram of this procedure. So the flow rates were 100 microliter per minute for the loading. So it was only about 3 minutes. After that, the flow rate for the washing procedure was raised to 500 microliter per minute. And so for the first washing step, here it switched to the second washing solution. And as you can see, about 80 microliter different from the column. It's the second wash volume. Arrived to the detector, and it was about 50 milliliter polyvolumes. It was flushed through the column. And finally, here in this area, the elution step, the first elution, and here the second elution step was performed, and fractions were collected. So this picture wanted to demonstrate the pressure profile from after loading of Hill digest on all 3 types of columns. back pressure in this case is much lower for the blank column, of course, because the particles there give a much higher back pressure, also from the nanoparticles compared to anode dioxide. So again, those collective fractions were CMS second dimension separated, and here I show you the first result for casing. Here you see the unique peptide spectral counts. Those are total numbers of peptides. On the left side, you see the fraction was put together to sample into the sample loading fraction. This contains closed interval fraction, and the elution fractions were comprehensive. And this means this is the column with the nano-titanium particles. In red, you can see the phosphorylated and the non-phosphorylated in blue. And as you can see, that the phosphorylated are much more counted in the elution fraction. And to compare this with the blank column, so you can see that most phosphopeptides which do not bind goes into the flow through and are not present anymore if you dilute. So this initially showed that the binding ability was fine. So the next step was to show the comparison of three columns after application of digest. So again, here you have spectral counts. These are relative values of phosphorated peptides on the left side now. In the load fraction in blue and the ALOR fraction in green. And here are these are the non-phosphorated from this is the blank column. If we compare this with N-titanium dioxide column, surprisingly there was no difference to the blank one. And compared to the column containing rutile nanodioxides, there was a huge improvement, so much more phospholates could be identified from the elution fraction of this column for phosphorated. Interestingly as well is if we see this part for the non-phosphorated peptides in the ALOR, we get these are now the absolute numbers of peptides found. There are quite a lot of non-phosphorated peptides there, which means that a lot of unspecific binding is still present there, and over the phase there still can be improved something in terms of flushing or washing the columns maybe. This is the same comparison on the protein level. On the left side, non-phosphorated, here the phosphorated ones in green, and so blank and this column, there's no difference for the phosphorated ones. 1.8-fold higher nanophosphorates were gained from this. So I want to start to summarize the first part. So the helix separation conditions for the Roman space were optimized and tested also with a complex biological sample digest, as shown here. The next step for this part should be to implement it online to perform a fully automated system. For the phosphate enrichment parts, it was shown that the column shows ability for binding phosphopeptides. There was no difference between the blank column and the hasty titanium dioxide column when prepping halo digest, also for peptides. And maybe this column can be checked by the separation guys if there's still some titanium dioxide particles are present. In contrast, the non-titanium dioxide particles should be 6-fold more